व्हाइट बोर्ड, जैसे व्हाइट बोर्ड दिख रहा हो, look at the white. Today's topic is a dominant epistasis, and this dominant epistasis is found has been this dominant epistasis has been reported in Cucurbita pepo. Cucurbita pepo is the botanical name of the pumpkin plant, and you know that in pumpkin plant three types of fruit color may be. The fruit color may be white, maybe yellow, and maybe green. Now look at, there are three genes. There are two dominant genes here. Look at that whiteboard. There are two dominant genes. The dominant gene W, the dominant gene which I have represented here by the alphabet W, and the dominant gene and the second non-allelic dominant gene I have represented here with the alphabet capital Y. So these are the dominant genes. The dominant gene, one dominant gene is responsible for the development of white color, and the another dominant is responsible for the development of yellow color. And this and the dominant gene for white color is epistatic to the dominant gene for the yellow color. And when this and the third color of the third types of the fruit color is the green, and the green phenotype appear when the recessive allele for these two dominant genes are present in the homozygous condition. I have written here small w, small w, small y, small y. This genotype will give rise to the production of green phenotype. What happens when abhi, just I have told you that each cross is made between two pumpkin plants, one having uh, one one producing white phenotype, another having green colored pumpkin fruit. What will happen in the F1 generation? In the F1 generation, all the plants 100% white phenotype will appear, and all the fruits and all the plants will produce the only white type. And when this F1 when plants when these F1 plants are allowed for the selfing to obtain the F2 generation, what will happen in the F2 generation? In F2 generation, we will find that at this structure, both the dominant allele both the dominant allele for white and yellow phenotypes are present there, but the dominant allele for white phenotype suppresses the expression of the dominant allele non-allelic gene for the yellow phenotype, and the color of the fruit will be white. And in this way, and when the dominant allele for white phenotype is absent and the dominant allele for yellow phenotype is present, the color of the fruit will be yellow. And when the recessive allele for both these dominant genes are present, green phenotype. Look at phenotype. Look at phenotype. When this in F, white, yellow, and green phenotypes appear in the ratio of 12 is to 3 is to 1. Anyway, this is the phenotypic ratio will be 12 is to 3 is to 1, but the genotypic ratio will be 1 is to 2 is to 1, 2 is to 4 is to 2, and 1 is to 3 is to 1. Yeah, what does it mean? It means that when a cross is made between the white and green 100% white phenotype is obtained due to the dominant epistasis here. This dominant allele W is the expression of the another non-allelic dominant gene, which is responsible for the development of yellow phenotype. And by suppressing the phenotypic expression of this dominant allele, all the plants will produce white phenotype. And when this F1 progeny are allowed for the selfing to produce the F2 progeny, in the F2 generation, the white, yellow, and green phenotype appear in the ratio of 12 is to 3 is to 1. So this is the ratio for the dominant epistasis is the ratio for the dominant epistasis is 12 is to 3 is to 1, and the ratio is 9:3. So today I have seen here the dominant epistasis. This is very important. Sangeeta, this is the dominant. Just I have shown you the dominant epistasis. Am I clear to you, Sangeeta?